So you probably already know that DNA is what makes up the genetic material. That means that genes are basically sequences of DNA nucleotides. Those get transcribed into messenger RNA, and then messenger RNA is used to make proteins through the process of translation. And we'll eventually explain how all those processes work. But it's important to go through a bit of the history because, again, it wasn't always well known that that DNA had this ability, or what even really the function of DNA was. In fact, for a long time, scientists assumed that proteins were the important genetic material. A given sequence of amino acids could take on a lot more structural properties than a given sequence of nucleotides, because again, for DNA or RNA, there are only 4 bases that you can use to make up a nucleic acid, whereas with proteins, there are 20 different amino acids you can use to make up a protein. So the assumption was that because you had more amino acids to use to make up a protein, there was a lot more information you could store in a given sequence or a given protein than in a given nucleic acid. So the assumption was that the proteins were what cells used or copied when they had offspring, and when parents had offspring, they were passing their genetic material in the form of protein. And in fact, it is the case that chromosomes, which are the structures that cells copy when they are going through cell division, for example, and when cells are passing their genetic material from one generation to the next, chromosomes themselves consist of both proteins and nucleic acids. So it's not entirely outlandish, or it wasn't entirely outlandish at the time, to assume that proteins were the things that were important for passing on genetic information. But we now know, of course, that DNA is in fact the genetic material. So it's worth looking back at how we now know that. So there are many experiments that kind of establish this, but some that are kind of more well known than others. So this experiment, carried out by Griffith back in 1928, involved using a strain of bacteria that was known to be deadly to mice. So the species of bacteria had 2 strains, one called the rough strain, one called the smooth strain. So they were the same species or same type of bacteria, just different strains. And the important difference between the two is that the rough strain was non-virulent, which means that if you injected it into a mouse, it would not cause any serious disease. Whereas the so-called smooth strain was virulent, which means that if you injected that into a mouse, it would cause disease, the mouse would die. So the question is, what part of the bacteria was killing the mouse? Now, again, it was known that everyone knew at the time that bacteria, just like other types of cells, had DNA and proteins. So the question is, what was it in these cells that was giving them this property? So, for example, you could take these smooth cells, these smooth bacteria, and heat kill them, which just means heat them up to boiling, and then inject that into a mouse, and the mouse survives. So the smooth strain cells have to be alive in order to cause disease, which means they have to be able to replicate. But then what Griffith showed is that if you mix together the heat-killed smooth strain cells, so just the components from those cells after they were killed, and then some rough strain cells, inject that into a mouse, the mouse dies. So what that means is that there's something in the smooth strain heat-killed cells which had the ability to transform the rough strain cells into the virulent S strain phenotype, which means that there was some information stored in those heat-killed cells that could transform the rough strain cells into virulent cells. So years later, this was the identity of those instructions, which we call the transforming factors, whatever it is that transforms the R strain and S strain cells was worked out by another group in 1944. And what they did is they took those heat-killed cells and separated the cellular components into the different major macromolecules. So using things like centrifugation and column chromatography, they separated the heat-killed cells into the protein component, nucleic acid component, the polysaccharides, and the lipids, and injected each of those into a live mouse with the rough strain non-virulent bacteria. And the only thing that could transform the rough strain cells into smooth strain cells was the DNA fraction. So again, this is 1944, which may seem like a long time, but we're talking about less than 100 years when the first sort of really definitive experiment showed that DNA was the substance that could cause a change, a transforming change in bacteria anyway. But there were still, at this time, a lot of people that doubted that DNA could contain all the information necessary for genetic transformation, again, because it only had these 4 bases. So a more definitive experiment came from these two, Hershey and Chase, in 1952, and they were looking at bacteriophages. So a bacteriophage is a virus that infects bacteria. So viruses, of course, are usually are considered non-living. They need a host to copy their genetic material and make new copies of the virus particles. And so a bacteriophage is just one that targets bacteria as a host. And so again, it was already known that these bacteriophages are very simple organisms. They only consist of protein and nucleic acid, so they don't have any lipids or any other kind of moving parts. So the only things that could be the transforming factor, the things that could carry the information necessary for making more viruses, either had to be in the nucleic acids or in the protein. So to figure out which it was, Hershey and Chase did an experiment where they grew viruses in a medium that contained either radioactive isotopes of sulfur or radioactive isotopes of phosphorus. And the reason they chose those 2 elements is that sulfur is found in proteins. So we know that sulfur can be part of at least 2 amino acids, right? Methionine and cysteine, but not in any nucleotide. So there are no nucleotides that normally contain sulfur. Phosphorus, meanwhile, is found in nucleotides. That's part of the phosphate group. But there are no amino acids that contain phosphates normally, unless they are phosphorylated after they're synthesized. But that doesn't happen with bacteriophage proteins. So by growing the virus in radioactively labeled radioactive isotopes of sulfur, the protein coat of the virus could be radioactive and therefore detectable, whereas growing the viruses in a medium that contained radioactive phosphorus meant that the DNA of the virus would be radioactive and therefore detectable. So in both cases, they took both strains, the sulfur-labeled viruses, the phosphorus-labeled viruses, and infected them into bacteria, 2 separate populations of bacteria, let the virus infect the cells, and then separate the infected bacteria from the whatever was left over from the virus, because the assumption was that whatever was actually entering into the bacterial cell contained the instructions for making more virus, whereas whatever was left behind presumably did not, was just a carrier molecule. It didn't contain instructions for making more virus. And so they let the radioactively labeled viruses infect the bacteria, and then separated them through centrifugation. So this is a simple way to separate things of different size. You put them in a centrifuge. Heavier things go to the bottom, lighter things stay at the top. The virus capsules were lighter than the bacterial cells, so this is an easy way to separate them. And then after separating them, look to see which fraction is radioactive. So when they did this with the radioactively labeled protein capsule, the ones that had radioactive sulfur, they found that the discarded part, the separated capsule, was radioactive, whereas the bacterial cells were not. On the other hand, when they did this with virus that had been grown in the presence of radioactive phosphorus, after centrifugation, it was the bacterial cells that were radioactive and the virus particles that were not. So that was kind of a definitive experiment that showed that DNA has to be the material, at least in the bacteriophage, that contains the instructions for making more virus, whereas the protein, at least in this case, is just a carrier for the DNA. And then other similar experiments showed that the exact same thing was true for pretty much, or for literally all living organisms. All organisms contain a sequence of DNA. And some viruses carry RNA instead of DNA. But for every other organism, it's a molecule of DNA, which contains the instructions for making proteins that are necessary for the survival and reproduction of the cell. So now we know that DNA is the genetic material. So the question is then, what is the structure of nucleic acid? And because, again, like with proteins, the structure of the molecule is going to be important for how it actually functions. So once it was established, again, we're talking about in the early 50s, that DNA must be the genetic material, the question became, what is its structure? Because that would tell us more about how it actually does this. Because again, there was still a mystery. How can DNA, which only has 4 different bases, contain all the information necessary for making all the proteins associated with an organism? So what we know first about DNA is that it can, or nucleotides anyway, can connect to each other through 5' 3' phosphodiester bonds. So when you want to connect two nucleotides together, you can connect, again, typically for most nucleotides, the phosphate is attached to the 5' carbon. And if you want to connect 2 together, you can attach the phosphate to the 3' carbon of another nucleotide. And that goes for both DNA and RNA. So the typical way that they're formed is in this 5' to 3' bond. And it's called a diester linkage because an oxygen with a phosphorus or another carbon on either end, and then connected to, double bonded to another oxygen, is an ester group. And so we call this a phosphodiester because it's an ester linkage between both nucleotides and the phosphate group. And you can, because the 5' and the 3' carbons are free on both ends, you can attach as many of these end to end as you want. And the base part does not really interact. So in other words, the base is not really involved in the phosphodiester linkage, which means you can have a sequence or a chain of these bases or nucleotides linked together in any order and for any arbitrary length that you want. And in fact, most nucleic acid molecules or chromosomes anyway, are millions and millions of bases long. So, and again, this goes for both DNA and RNA. One important thing is that with a chain like this, it always has kind of a polarity. So the end that has a 5' free, in other words, whichever end of the strand has nothing attached to the 5' end other than a phosphate, we call the 5' end of that chain. Whereas the other end we call the 3' end. Again, the analogy is with proteins. So a chain of protein or amino acids linked together with peptide bonds has an amino terminal and a carboxy terminal. And so with a strand of DNA or RNA, we call one end the 5' end and one end the 3' end. Now the structure of a DNA molecule, again, can be at the same time more complicated and simpler than that of a protein. So with a peptide bond, there you have a central carbon atom with 2 two bonds linking it to its adjacent partners, two peptide bonds on either side, and the peptide bond itself doesn't rotate, which means there are actually there's not that many configurations or a limited number of configurations that a peptide bond can take on. But with the phosphodiester linkage, there are many bonds that are able to rotate. So there are at least 7 different bonds that have the ability to rotate. The bonds from the phosphate to their oxygen atoms, and then the linkage to the 5' and 3' carbons. The connection between the 5' carbon and the 4' carbon of the ribose molecule, and even the bond between the 4', so that's this bond here, and then the bond between the 4' and the 3' carbon can rotate, although it's part of a ring, so it can only go between that endo and exo configuration. And then there's the bond between the 1' carbon and the base, which is able to rotate. So that means there's a lot of degrees of freedom. Now with the bond between the base and the ribose, it depends on the base. So with adenosine, for example, or with purines, for example, they can, because of steric interference with the ribose, what's called the syn configuration, where the ribose, the base is sort of oriented over the ribose, is less stable than the anti configuration. Whereas with pyrimidine base, it can go between the syn and the anti configuration more easily. But in any case, there's lots of different ways in which these bonds can rotate, which means there's lots of potential structures that a nucleic acid molecule could take on. This is why it was so difficult to work out the structure of DNA. So in fact, while the structure of DNA was being worked out, the 3-dimensional structure of proteins was being worked out. So things like the alpha helix and the beta sheet of proteins, those had been already pretty well established by the time people started looking more in depth into the structure of nucleic acids. So there were a lot of different groups all over the world who were trying to work out the structure of DNA, because again, the assumption was if you could figure out the structure of a DNA molecule, you could understand better its function, so how a DNA molecule could act as the genetic material. And there were lots of different groups trying to figure this out in lots of different ways. One important person that was really one of the people that really figured out or did one of the key experiments in figuring this out was this lady, Rosalind Franklin. And so she worked at Oxford University with under this guy, Maurice Wilkins. And the two of them were doing X-ray diffraction. So we talked about X-ray diffraction in the context of protein structure, but you can use X-ray diffraction hypothetically for just about any kind of molecule. DNA is no exception, and so but X-ray diffraction is very difficult, very time consuming. You have to have a very pure source of your molecule. You have to get it to crystallize properly, which is hard to do. And then you take the crystal of your molecule and again pass an X-ray beam through it. The diffraction of the beam onto your photographic film will give you different patterns depending on the structure. So when Rosalind Franklin did this, she got this this kind of now famous X-shaped pattern here. Again, you have to really understand X-ray diffraction. You have to know the precise geometry of the X-rays and how they hit the film in order to interpret it. But basically, Franklin knew from this pattern that the structure must have some sort of helical nature, meaning some sort of spiral-like shape. So that was an important clue for the structure of DNA. Another important clue came from a guy named Chargaff. And Chargaff wasn't actually so much interested in the structure of DNA, but in the makeup of the bases of any given DNA molecule. So Chargaff looked at the DNA samples from lots of different sources and looked at the amount of each of the different bases. So, and when he did this, he found some strange things, or strange at the time anyway. So, for one thing, he found that whenever he measured the concentration or the relative amounts of purines, for example, in a sample compared to the number of pyrimidines in a given sample of DNA, they were always the same. So, the percentage of a sample that was purine, or every sample was always about 50% purines and 50% pyrimidines. And stranger still, whenever he looked at the relative amounts of each of the bases, he found that the percentage of any given DNA sample from a cell that was adenine or adenosine would be the same, equal to the percentage that was thymidine. And then the percentage guanosine would be equal to the percentage of cytidine. And then on top of that, if you look at the relative proportions of these bases within an organism, they would be the same. So in other words, if you compare the percent A in, say, liver tissue from an animal, it would be the same if you looked at, say, a piece of brain tissue or a piece of skin tissue. And not only that, but it would be the same within species. So if you looked at two different members of the same species, the relative makeup of each of these bases would be the same. But they would be different across species. And this was weird. No other molecule shows this kind of behavior. So for example, if you look at the amino acid content in a given tissue sample, the relative amount of, say, glycine versus tryptophan, for example, will vary from tissue to tissue because different tissues express different numbers of proteins. And any given protein could have any given combination of amino acids. And the same is true for other types of molecules as well. If you look at the makeup of monosaccharides or lipid molecules, you never see these really precise ratios like this. So these combinations were another clue that there's something about the pairing of adenine and thymine and the pairing of guanosine and cytosine that must tell us something about the structure of DNA. And in fact, these are called Char-gas rules. So the fact that the relative amount of adenine in a DNA sample is always equal to the relative amount of thymidine, the relative amount of guanosine is always equal to the relative amount of cytosine, and that those ratios are consistent across tissues within an organism and consistent across individuals of the same species. So this relationship between adenine and thymine and between guanine and cytosine comes from the fact that those bases have the ability to form very precise linear hydrogen bonds, and when they're paired this way, we call them base pairs. So with adenine and thymine, they each can form two hydrogen bonds with each other. So there's a pair that forms here, a hydrogen bond that forms here in a nice straight line, and one that forms between this amino group and this carbonyl group, carbonyl oxygen of thymine. Meanwhile, cytosine, if you have a molecule of cytosine and a molecule of guanine next to each other, they also form hydrogen bonds. They form 3 instead of 2. So there's a bond here between this amino and this carbonyl oxygen, between this amino group and this nitrogen, and this amino group and this carbonyl oxygen. So they form these very nice parallel hydrogen bonds, which are very stable. And a couple of things. One, they only form hydrogen bonds this way. So in other words, if you try to form a base pair between, let's say, a cytosine and an adenine, they don't pair up because the positions of those amino groups and those carbonyl groups don't line up the way they do if you try to pair a cytosine with a guanine or a thymine with an adenine. Also, the distance between the hydrogen bonds is very consistent. So, if you compare just the distance from the 1' carbons of an adenine and a thymine, so that's where they bind to the ribose group, the distance is almost exactly the same. So it's 11.1 angstroms between an adenine and a thymine, and 10.8 angstroms between cytosine and guanine. So again, another clue that this base pairing is probably why Char-gas rules apply, and so the structure of DNA must somehow incorporate this this base pairing. Then these two guys, James Watson and Francis Crick, took what they knew about DNA. So knowing how the linear nature of nucleotides linked together through phosphodiester bonds are formed, knowing Char-gas rules, and knowing they actually worked with or in the same at the same university as Rosalind Franklin and Maurice Wilkins, and they looked at Rosalind Franklin's X-ray diffraction data, and they used that to come up with the DNA double helix. Now, there's some debate about whether they actually were, or Rosalind Franklin gave them her data so that they could come up with this structure, or whether they kind of stole it. History's kind of still, or jury's kind of still out on that, but in any case, they were the ones that came up with the now famous double helix structure of DNA. Because if you have two strands of nucleic acids, if they are complementary, and that means that they have bases on opposite strands that match in this Char-Gaff-like system. So if you have a cytosine on one strand and then a guanine on another strand of the same position, those two will hydrogen bond, and then an adenine in the next position, then there has to be a thymidine in the next position on the opposite strand. As long as both strands are complementary, then they will hydrogen bond with each other and come together like two sides of a zipper, sort of. Not only that, but the base pairs, or when the bases are paired like this, because both molecules are basically flat. So both pyrimidines and purines are basically flat molecules when they're, because of all the double bonds that hold them together. Two bases that are base paired will also be flat. So the two strands can line up because are with each base pair almost parallel to the one above it, and that's true at each nucleotide sequence. So that's why you have this almost spiral staircase like arrangement with each base pair basically lying in a completely two-dimensional plane. And then the nature of the phosphodiester bond allows the strands or the strand to turn slightly, and so that gives rise to the helical nature of the structure. Importantly, like I said, the bases are not part of the phosphodiester linkage. So the phosphodiester linkages are always on the outside. So this is the phosphodiester linkage going around the outside of each strand. The bases, therefore, are always on the inside, base pairing with each other. And again, because that distance between the bases is always pretty much the same, the width of the double helix is pretty consistent all the way down, or the shape of the double helix is consistent regardless of the sequence. So it does not matter what bases are present at each of these positions. The width of the double helix is pretty much always the same. The degree of rotation of the helix is always the same, going all the way up the molecule. So the two strands kind of wrap around each other in a not quite symmetrical way, so you end up with two grooves, what's called the major groove and the minor groove. It's a little bit easier to see actually in this view. So here, in fact, I'll make it a different color. So here would be the minor groove, and here would be the major groove. And so each one, again, wraps around the double helix all the way from end to end. The period, meaning the distance from that a strand takes to make one turn, is 36 angstroms. So that's how long it takes to get from to make one complete turn. And the width of the helix is about 20 angstroms, and the distance between bases is about 3.4 angstrom. So it forms a very regular structure, again, regardless of the base sequence. So that's why you can have, as long as the two strands are complementary, they will always have that same structure. One important thing, though, that I didn't mention is that the two strands, in order to form the double helix, have to be anti-parallel. And so what that means is that the two strands have to be positioned in opposite orientations. So if this is the 5' end of one strand, that means that this is the free 5' carbon of that strand. It has to correspond to the 3' carbon, or the 3' end of the other strand, and vice versa. So you have one strand oriented in the 5' to 3' direction. The other one has to be oriented the other way, with respect to the first one. And so when that is the case, we say that these two strands are anti-parallel. And so DNA and RNA can only form this double helix structure when when the two strands are anti-parallel to each other. And so Watson and Crick shared the Nobel Prize for this discovery with Maurice Wilkins, who again worked with Rosalind Franklin. Now Rosalind Franklin perhaps would have gotten the Nobel Prize too. Unfortunately, she died in 1958 from ovarian cancer before the Nobel Prize had been awarded. There is a rule, the Nobel Prize Committee has a rule that it cannot be awarded posthumously, and that it can only be split among 3 people. So the award went to Watson and Crick and Wilkins. Again, the controversy that still rages today is whether or not Rosalind Franklin would have gotten the Nobel Prize if she had lived. But again, she is the one that did the work that established that there must be a helical structure. In fact, Watson and Crick themselves didn't really do any experimental work at all. They did all of their work just with molecular models. Now, of course, this was back when they did everything with paper and pencil, and they had 3-dimensional plastic and metal models that they could work with long before there were computers that could work out the 3-dimensional structure of complex molecules that we have now. But they worked out the structure almost entirely as sort of a thought experiment. So they didn't do a lot of actual experimental work, unlike Rosalind Franklin. But in any case, the structure is important because it does tell us some things about how DNA works. So this complementary anti-parallel structure is the secret of how, for example, DNA can replicate. Because we know, of course, that DNA, in order to be the genetic material, it has to be able to do 2 things. It has to be able to encode the information necessary for making proteins and therefore carrying out the functions of the cell. And it has to be able to be copied in the process of replication, because when cells divide, each daughter cell has to have a complete copy of its genetic code. And when an individual produces offspring, the offspring also have to have a complete copy of the parent's DNA in order to keep the DNA propagating. So this again, complementary nature of the 2 strands is the clue for how that's possible. Because if you have 2 strands that are complementary, that means you can separate them, and you can always tell what the other strand was from one of the parent strands. And this is in fact exactly how DNA replication works. Now later on we'll learn how DNA replication works in more detail, but the basic idea is that you have a parent molecule which consists of 2 strands. Those are separated, and then an enzyme that's actually called DNA polymerase, because it makes a new copy of DNA, reads the parent strands basically as a template and starts making the daughter strands. And it's able to do that because, again, it knows the enzyme can match the new daughter strand sequence to the parent strand sequence. So, for example, if it sees, for example, an A in the parent strand, it can add a T to the daughter strand, and so on. And so that complementary structure is and being anti-parallel also is important for replication. And so later on we'll talk about how that works, and we'll also talk about how the process of actually using DNA to encode proteins works as well. Now we now know that DNA structure is a little more complicated than, or can be more complicated than what Watson and Crick came up with. In fact, what the structure that Watson and Crick discovered, we now call the B form of DNA, and it is one of actually 3 different structures that DNA can take. The other 2 are called the A form and the Z form, and so you can kind of see each one here. The A form is tighter. In other words, the distance between turns is shorter, so it's 28 angstroms instead of 36. And the bases, while still base pairing and still complementary, are arranged kind of around a almost hollow core, whereas in the B form DNA, the bases are almost directly over the center of the helix. And then the Z form is even looser and a little more complex, because the bases still are base pairing with each other, but the way in which the riboses are oriented and the backbone is not quite as smooth. Also, there's a difference in the helix. So both A and B forms of DNA are what we call right-handed helices. So that means that if you were to kind of wrap your hand around the helix with your thumb facing up, you can only do that with your right hand. Whereas with Z DNA, it kind of goes the other way. So if you wrap your left hand around actually I should draw the arrows this way, your left hand around the Z form DNA helix, then your thumb would be facing up in this particular diagram. And so this just kind of shows the different parameters for different structures. Again, A and B are both right-handed. B is a little bit narrower. A is the widest of the 3 in terms of diameter. The number of bases per turn is about the same for all 3, but the rise per base is pretty similar. The only other difference is the base tilt. So in other words, for a B form DNA and Z form, the base tilt is very small, whereas for A form, you can kind of see here when we say base tilt, it's kind of this angle here relative to the long axis of the helix. So for B and Z, the bases are almost perpendicular to the long axis of the helix. And then there's differences in how that ribose molecule is oriented. So for A form, it's most of them are are in the endo configuration, 3' endo configuration, whereas for B form, it's the 2' endo configuration. And then for Z, it can vary depending on if you're talking about pyrimidines or purines. And then again, the glycosyl bond conformation, that's the conformation of the bond between the base and the ribose is always in the anti-conformation for A and B. But then again, for Z form DNA, it can be either anti or syn. So the Z form DNA is more complex. Now, the B form is the important one, because that is how most of the DNA in a cell and most of the time will be found. But A form and Z form forms can appear under certain circumstances. So depending on things like water content, for example. So they are important, but B form is the most common form of DNA. And other structures besides the double helix can show up. I said before that the sequence of bases in a DNA molecule does not affect the structure, but that's not always true. There are certain sequences of bases that can give you different types of structure. So And one of those are called palindrome sequences. So you probably know what a palindrome is. It's a word or a sentence that that reads the same forward and backward. So like taco cat, for example, is a palindrome because you can read it forwards or backwards and it reads the same way. But with nucleic acid sequences, a palindrome is not necessarily the same sequence forward and backward on one strand. That would be a mirror repeat. So this is a mirror repeat, a sequence like this. So TTA, GCAC, and then CAC, GATT. That's a mirror repeat. A palindrome is where the sequence is the same on reading on from either end on the other strand. So this sequence TTA, GCAC, GTG, CTAA is a palindrome because that same sequence shows up on the other strand if you start at the other end. And remember that these are anti-parallel. So if this is the 5' end of this strand, that means this is the 3' end, and this is then the the 3' end of the other strand, and this corresponds to the 5' end. So again, if you start at the 5' end of either strand, the sequence is the same. So that's why we call this a palindrome. Also, by the way, one bit of convention I forgot to mention, whenever you write nucleotide sequences, it's conventional to write the 5' end at the beginning. So the 5' end is always at the left, and then the 3' end is usually at the right, unless you're writing two sequences that are complementary to each other, in which case, of course, one end has to be the 5' end, and the other, that same end, has to be the 3' end for the other strand. And so, but then again, by convention, the top strand is written with the 5' end at the left and the 3' end to the right. So these palindrome sequences are unique because they can form what are called hairpin structures. So because the one end, the 5' end of a palindrome sequence is the same as the 5' end of the other strand, that means the 5' end of a palindromic sequence is complementary to the 3' end of the same sequence. So again, this is a palindrome sequence. So this TGC GAT is, if it shows up, if this sequence down here shows up ATC GCA, that is sort of self-complementary, which means that this strand can form this hairpin loop where part of this sequence, this region here, is complementary to itself, with about 4 bases in between that are not complementary and don't base pair. Now there's a limit to how short this can be. If it's any less than 3 or 4, this bend is too tight for this structure to form. But if you have 2 complementary sequences like this in the same strand, they can form this hairpin loop. And if you have 2 strands and they're both palindromic, then both of them can form hairpins, and that's called a cruciform structure. Cruciform means cross-like because essentially that's what it looks like, is a big cross or plus sign. And these structures are important. They show up in certain regions. There are a lot of protein or DNA interacting proteins that bind to or interact with these hairpin or cruciform structures. And then there are even more complicated structures that can form with special DNA sequences. So you can actually form triple or quadruple helices. So that means a triple helix would be 3 strands that kind of wrap around each other, but only with certain base compositions. So if you have this arrangement, for example, where you have a thymidine sort of in a standard base pairing with an adenine, it can then form actually another set of hydrogen bonds with another thymidine this way. And so if you have 3 strands that have adenines and thymines in them in the right proportion, they can form triple helices. Same way with C and G. So this is a typical CG base pair, but then that guanine can also form 2 hydrogen bonds with another cytosine. It's not as stable, but again, if you have 3 strands that are just C's and G's in the right order, then you can form a triple helix. So again, these don't form very often in practice or in real cells because the right combination of bases doesn't show up very much, but you can create these artificially. You can even form a quadruple helix if you have 4 strands of DNA that only have guanosine bases, because guanosines can hydrogen bond with other guanosines, but only if they're arranged in this sort of tetraplex complex. So if you have, again, 4 strands of DNA that just have guanosine, you can form a quadruple helix. But again, those don't show up in nature. They only really show up in artificial setups. Now, RNA, on the other hand, can form a lot more complicated structures than DNA, simply because, again, that missing hydroxyl group, it's able to take on more complicated conformations. And in fact, typically, RNA is only found in its single-stranded form. So whereas DNA in most cells most of the time is entirely double-stranded, RNA is typically only found in single-stranded form. And so single-stranded RNA can form lots of, because of the ability to form base pairs with itself. If it's self-complementary, you can see lots of complex structures. So a single RNA can form lots of hairpin helices, and even when you have 2 strands that are together, it's a lot more forgiving of places where there isn't good base pairing. So you can see these loops and bulges, which are places where you have mismatches or positions where there is no base for a given sequence to pair with. So that's why RNA is able to take on a lot more complex structure. RNA, therefore, molecules can form things like this actually is a transfer RNA here, and so it has a very complex 3-dimensional shape, which does not look at all like the DNA double helix. And so, again, you see hairpins in there, and you see lots of places where you can have unpaired bases, and even within a molecule like tRNA, you can see a lot of sort of non-standard base pairing occurring. So these are all here. These are all combinations of bases that occur within a transfer RNA molecule. And then over here, there are just other more complex RNA structures. In fact, again, it's worth mentioning the parallels between RNA and proteins. So RNA molecules can form very large complex structures, kind of like proteins, and that's one of the reasons why it's thought that RNA molecules might have been the precursors to proteins, because RNA molecules some RNA molecules actually can act as enzymes. They're called ribozymes. So certain types of enzymatic reactions require RNA molecules instead of proteins. And so it's thought, and because therefore RNA has the ability both to store information and to act as enzymes in the right context, it's thought that maybe the earliest life forms, or the precursors to early life forms, consisted entirely of RNA molecules, and did not contain proteins and DNA until later. All right, so next time we will talk about the chemistry of nucleic acids.